Hello and welcome to my YouTube channel, AnatomyMentor. And today we'll be discussing the very important topic of anatomy, which is the histology of the stomach. Now, this is a topic which has been often included in exams, and it is also having a lot of clinical relevance. We will be discussing peptic ulcers at the end. But in the beginning, we shall talk about the basic histology of the stomach, and we will discuss the gastroesophageal junction as well as the histological characteristics of the layers of the stomach wall. And we will also discuss a label diagram in which I'll show you the general morphology of a gastric gland and location of the gastric gland cells. And I shall also show you the different regions of the stomach on the basis of the histology. As well as that, you can also be familiar with the cells which are present within the mucosa of the stomach. And we shall talk about the clinical conditions. Like I told you, this is a clinically relevant and important topic, that is the peptic ulcer. And we shall also discuss the role of Helicobacter pylori, the bacterium, in peptic ulcer. So these are learning objectives for today. And as I start with the topic of histology of the stomach, I like to show you, first of all, that what you should be able to identify is the gastroesophageal junction. So over here in front of you, you can see that this here is the gastroesophageal junction. And you can appreciate that over here, the epithelium, it has changed from the stratified squamous epithelium in the esophagus to the simple columnar epithelium within the stomach wall. So when we're looking at a histomicrograph of the gastroesophageal junction, we see this change in epithelium is abrupt. This is not a gradual change. This is an abrupt transition of the epithelium. So the stratified squamous non-keratinized epithelium becomes simple columnar. And also we should note that there are cardiac glands in the esophagus. In which part of the esophagus? That is the innermost mucosal layer, specifically in the lamina propria of the mucosal layer. And these cardiac glands, they secrete neutral mucus, which protects the esophagus from the acidic secretions of the stomach. Then let's look at over here the part of the stomach, which is adjacent to esophagus. Here also in the mucosa, the cardiac glands are present in the lamina propria. So you can see these glands easily. The rest of the layers are the typical layers which are present in the gastrointestinal tract. Below the mucosa, you can see that there is going to be the submucosa and then the muscularis externa, and external to that will be the serosa. Here is another histomicrograph which is showing us a much better clear aspect, where we can see this here is the stratified squamous non-keratinized epithelium of the esophagus. And here on the left of this slide, it has abruptly changed into the simple columnar epithelium, which is the innermost lining of the mucosa of the stomach. We also appreciate the other layers in the stomach wall. That is the lamina propria, that is outer to this here columnar epithelium, and then muscularis mucosae. Collectively, the epithelium, lamina propria, and muscularis mucosae are known as the mucosa of the stomach. And here the lymphoid tissue, you can also see that. And over here then comes the submucosa, that is external to the mucosa. And then external to that is the muscular layer. What are the functions of the stomach if we want to summarize them? Well, we know that this is the place where the food is going to be mixed with the gastric acid and the other secretions, and it will be eventually reduced to a fluid mass, which is known as chyme. Now, the digestion of carbohydrates, remember, begins in the mouth by the enzyme amylase, which is present in the saliva. So this process of digestion of carbohydrates continues within the stomach. Also in the stomach begins the digestion of the fats or triglycerides by the enzyme, which is the lipase. And the initial digestion of proteins by the enzyme pepsin will also begin in the stomach. So here is a very simple diagram which is showing us the different parts of the stomach. And we can see that this here is the esophagus, which is leading into this pouch-like organ, which is the stomach. and the part of the stomach which is above the entrance of the esophagus, this is known as the fundus of the stomach. And the point where the esophagus enters the stomach, this area is known as the cardia. The rest of the stomach, the major portion, is known as the corpus or the body of the stomach, and inferiorly it narrows into this area which is called the pylorus. And at the pyloric sphincter separates the stomach from the duodenum. Here we can see in this drawing these longitudinal folds, which are known as the rugae. They comprise the innermost layers, that is the mucosa and submucosa of the stomach. And these longitudinal folds, they facilitate the distension of the stomach when it is filled with food. Okay, so we have talked about the 4 basic layers present within the stomach wall before, and I'm going to summarize these layers. There is a general pattern I told you from the inner to the outer aspect within the gastrointestinal tract. There are 4 layers histologically speaking. That is the mucosa, submucosa, muscularis externa, and then the outermost, which is the serosa or adventitia. So the stomach also has these 4 layers. In case of the stomach, the mucosa has its further 3 constituents from the inner to outer aspect. These are the epithelium, lamina propria, and the muscularis mucosae. I told you the epithelium is the simple columnar epithelium, meaning the cells over here are tall, columnar, with a nucleus which is oval-shaped and in the basal region. The lamina propria is simply the loose connective tissue, and this is a thin and delicate layer which is supporting the epithelium. And then comes the muscularis mucosae, which has a few smooth muscle fibers, and they facilitate the secretions of the epithelium and the slight movements of the epithelium that take place. Now, if we look at the submucosa, this is another connective tissue layer which is supporting the mucosa, and then outer to that is the layer which has the thick quantity of the smooth muscle fibers, that is muscularis externa or propria. So this is the proper muscular layer of the stomach. And in case of the stomach, the muscle fibers, the smooth muscle fibers, are arranged in 3 layers. So based on the direction of these muscle fibers, there is an inner oblique layer, a middle circular layer, and an outer longitudinal layer. Then the most external one, in case of the stomach, it is the serosa, which is, you could see, a thin lining of squamous cells, and that is the flattened cells supported by delicate connective tissue. This is a very simple diagrammatic representation of the 4 layers of the stomach, which I just discussed with you. So the innermost layer here, this one is the mucosa. Now, its constituents are simple columnar epithelium, below that the lamina propria, and here you can see the continuous muscularis mucosae. Like I told you, the simple columnar epithelium consists of the cells which are tall and columnar, and these cells, they invaginate into the underlying lamina propria, and this is how they produce the glands. So it's simply the invagination of the surface layer into the underlying lamina propria, which are known as the glands, and the point of invagination is known as the gastric pit. We here see the lamina propria, which has blood vessels and lymphocytes, and then beneath that, this here is the muscularis mucosae. I told you the function of muscularis mucosae is that it is responsible for the slight movements of the epithelial layer and also the secretions which are going to be produced from these gastric glands and pouring those secretions into the stomach lumen. So that is the function of muscularis mucosae. Now, the mucosa is supported beneath by the submucosa, which mainly consists of the blood vessels, and it also consists of the submucosal nerve plexus. Submucosal nerve plexus is responsible for the supply, the nerve supply of this here muscularis mucosae. Now, beneath that comes the main muscularis propria, consisting of those 3 layers of smooth muscle fibers, inner oblique, middle circular, and the outer longitudinal layer. And this is supplied by the nerve plexus, which is the myenteric nerve plexus, and also known as the Orbach's nerve plexus. So this is the one which is responsible for the movements, the churning movements of the stomach as a whole, which turns the food into the chyme. Outermost layer you can see is the serosa, consisting of the flattened squamous cells. So these are the 4 layers present in the wall of the muscle as depicted by a simple drawing or a diagram. Now we can understand the micrographs since we have understood the basic layers of the stomach. This is a photomicrograph of the stomach wall fundus at low power, and this is stained by hematoxylin and eosine. Over here, we can see the mucosal layer, which has been labeled as M over here. The mucosal layer has this epithelium, which is then invaginating and forming the glands. And between the glands is lamina propria, and beneath lamina propria here, where you can see my laser pointer right now, this is showing us the muscularis mucosae. Collectively, these three layers are known as the mucosa of the stomach. And beneath this mucosa is then this submucosa labeled SM, and it has the blood vessels, and it has also, you can see the delicate tissue fibers. So these two layers, mucosa and submucosa, they are thrown into these larger folds, and these larger folds of the stomach, remember, are the longitudinal folds known as the rugae, which are visible on gross examination, or we could say they're visible to the naked eye. Let's look over here. Beneath the submucosa is the muscularis externa, and these are the muscles arranged in different layers, in different directions. And eventually, this is the serosa on the most outer aspect of the stomach wall. So remember, the epithelium of the mucosa invaginates deeply into lamina propria, forming the gastric pits. And the gastric pits, now this is an important point to remember, the gastric pits, I told you, as they invaginate the lamina propria, they form the gastric glands. Now these glands are simple branched tubular glands. Now if you recall from my lecture on the different types of glandular epithelium, and when we talked about the classification of the glands, when we use the word simple, then what we mean is that the conducting portion of the gland or the duct of the gland is single and it is unbranched. But when we use this word branched tubular glands, then we are talking about the secretory portion of the gland. The secretory portion of the gland is branched and it is in the form of tubes. So that is why these are known as the simple branched tubular glands. So here now we're going to focus on the gastric glands, and this is a section, a histomicrograph, which has been stained by the special stain alcine blue and periodic acid shift stain. And here you can see the simple branch tubular glands in the fundus of the stomach. So here we're going to look at this portion over here. So remember, I told you that the point of invagination of the epithelium at the lumen of the stomach, that is known as the gastric pit. And then this epithelium, it invaginates, and you can see then it forms these deeper lying gastric glands. So the gastric gland itself, it actually starts from the bottom of the gastric pit. This is the gastric pit, and here right now I have fixed the laser pointer at the region which is the bottom of the gastric pit. So this is the point from where the gastric gland actually begins, and then it extends all the way down to the muscularis mucosae. So this portion is now the gastric gland. And what are the parts or subdivisions of the gastric gland further? Well, this here opening is known as the isthmus, which literally means mouth. And below that isthmus is then the region known as the neck of the gastric gland. And the lowermost part of the basal region is also known as the fundus of the gastric gland. The fundus, you can see, is wider in comparison to the neck, which is narrow. Now, the whole of this gastric gland, remember, is lined by the epithelial cells, of course, but these are different types of epithelial cells, and we will be talking about them in the further slides. So each cell has its own distinct function, its distinct histology. Those ones which are present at the surface of the gastric mucosa, those are known as surface mucous cells. Surface mucous cells, I told you before, simple columnar cells with the basal oval nuclei. And you can see they have been here pointed out by the black arrows in this histomicrograph, and they line the surface epithelium, so they also extend into the gastric pits. Their function is that they secrete the mucus, which forms a thick, lubricant layer on the inner surface of the stomach to protect the stomach itself from the erosive action of the the gastric juices, the hydrochloric acid, which is the secretion of the cells of the stomach. The gastric glands, they also contain other cells. Now, what are the other cells which are present within the glands? Well, within the gastric glands, we have the mucous neck cells. And mucous neck cells, remember, are different from the surface mucous cells, which I just showed you, which were lining the gastric pits. The other types of cells within the gastric glands are stem cells, parietal cells, also known as oxyntic cells, the chief or zymogenic cells, and enteroendocrine cells. Now, each cell type has its specific property that it has a unique characteristic, and it also stains differently. Now, since we're going to talk about these cells individually, first I'm going to show you a labeled diagram depicting the different cell types, a label diagram of the gastric gland. So here, when we talk about the gastric pit, we know this is the region which is lined by surface mucous cells. Surface mucous cells are the cells in simple columnar epithelium, and these are the ones which are continuous from the inner aspect of the lumen, that is the surface cells. They are present within this epithelium. And when the actual gastric gland begins at the isthmus, then we can see now the other cell populations appearing. Amongst these are the mucous neck cells. They are slightly different from the surface mucous cells. And also we can see these prominent pink cells, these large cells over here, which have been labeled the parietal cells or oxyntic cells. Let's go further down in the gastric pits. We will see in the fundic region of the gastric glands. In the fundic region of the gastric glands, we can see over here, we now have the enteroendocrine cells, and we also have the chief or zymogenic cells. So these are the different cell types which are present within the gastric glands. The 5 cell types: mucous neck cells, parietal cells, enteroendocrine cells, and the chief cells. And the 5th one, that is the stem cells as well. Okay, so mucous neck cells, what makes them different from the surface mucous cells? Well, first we remember that they are present not within the gastric pits, but they are present within the gastric glands. So they are interspersed among the parietal cells. In comparison to the surface mucous cells, these cells are less columnar. Their nucleus is of a rounder shape in comparison to the nuclei of surface mucous cells. So here is the stomach, H&E, hematoxin eosine, plus the special stain. And over here, we can see that these are the prominent parietal cells. They're larger, and in between them, we can see here are the mucous neck cells. Now, mucous neck cells, they are also secreting mucus like the surface mucous cells. But the difference is that their mucus is less alkaline, soluble mucus, compared to the surface mucous cells, which produce a highly alkaline, cloudy, or insoluble mucus. So for the same reason, the mucus cup is less prominent in case of the mucous neck cells. The stem cells, as the name shows, these are the cells which are responsible for the generation of the other cells. So they're progenitor cells for all the other cell types, and they are mainly present within the isthmus of the gastric gland. Surface mucous cells, they are, remember, renewed approximately every 3 to 5 days. Parietal cells and chief cells, these two cell populations, you can see them over here in the histomicrograph. Parietal cells have been labeled by the blue arrows. You can see that they are the eosinophilic, that is the pinkish stained cells, and they have a characteristic fried egg appearance, and these cells are present more often in the neck of the gastric gland. But over here, we can also see the cells which have been pointed out by the black arrows, and there you can see the purplish bluish staining. So they are having the basophilic basal region is especially basophilic. These are the chief or zymogen cells, and they're found in the lower parts, the deepest parts of the gastric glands. Now, what makes these chief cells basophilic is because of their large quantity of rough endoplasmic reticulum, whereas in the apical region of the chief cells, there is a slight eosinophilia because of the zymogen granules that contain enzyme precursors. So what are the chief cells producing? What is their function? They are producing pepsinogen. Pepsinogen is a substance which is converted into its active form pepsin within the acidic environment of the stomach. And these pepsins are then responsible for the breakdown or hydrolysis of proteins. Gastric lipase also, which is going to be present, is going to be produced by the chief cells, which is responsible for digestion of lipids. So here we can see the diagram in which we are looking at then again, we're looking at a gastric gland. The upper part is the gastric pit, and then in the lower part, you can see this here gastric gland. And the different cell populations, like I told you, these are the surface mucous cells which are present in the inner lining of the stomach wall and also in the region of the gastric pit. This is the cell type which is secreting mucus. And then as we come to the region of the gastric gland, we can see in its isthmus, you can see this here is the mucus neck cell, which is also secreting mucus. But like I said, this mucus is less alkaline in comparison to the mucus which is produced by the surface mucous cells. Then we can see these prominent parietal cells. Now, these are the cells which are secreting hydrochloric acid and intrinsic factor. And then in the lowermost portion of these gastric glands are the chief cells, which are producing the pepsinogen and lipase, and also the enteroendocrine cells. In this case, they are the G cells producing the hormone gastrin. So we will be talking about enteroendocrine cells in a short while. First, let's look at the parietal cells. So the function of parietal cells is now being clear to you, that the function is that they are producing an intrinsic factor, a glycoprotein that complexes with vitamin B12 in the stomach and the duodenum. And then it's necessary for the absorption of this vitamin B12 in the ileum. The other substance, very important substance, which is produced by parietal cells is the hydrochloric acid. Hydrochloric acid is the substance which gives gastric acid, juice acidic pH, that is a low pH, about less than 1 to 2. And this is then also responsible for digestion of dietary protein by acid hydrolysis of the substrates. And it also converts pepsinogen into pepsin. Plus, it is also bacteriostatic. So bacteria would die, or the growth would be inhibited in the acidic environment of the stomach. Let's talk about the clinical condition pernicious anemia, as we are discussing over here the function of the parietal cells. Damage to these parietal cells can occur in case of an autoimmune disorder or some other reason, and that can eventually lead to a deficiency of the intrinsic factor. Consequently, vitamin B12 will not be absorbed, which was the cofactor needed for DNA synthesis. And this causes the reduced proliferation of erythroblasts, producing the anemia. Now, coming back to the basic histology and function of parietal cells, remember that parietal cells can be present within two states, either the active or the resting state. Now, in the active state, a parietal cell will have large quantities of mitochondria, abundant microvilli protruding into intracellular canaliculi. So this is a specific feature of parietal cells. When you look at these cells in the microscope, you see that they have these invaginations known as the canaliculi, and you've got microvilli protruding into the canaliculi. But if the parietal cells are in the resting state, the number of mitochondria will be less, and the cells will have fewer microvilli, shorter intracellular canaliculi, and instead of the intracellular canaliculi, tubular vesicles will be seen. So let's look at that in a simple diagram. If you're looking at the ultrastructure, that is the magnified version of the parietal cell, and we want to compare the difference between resting and active state, then we can see over here that on the right side, we can see the active state of parietal cell, in which there are the large number of mitochondria. And here is the intracellular canalicus. This is, you can see the invagination, and within this, you can see are projecting lots of microvilli. Let's compare it on the other side, that in case of the resting state, you can see the number of mitochondria is less, and the intracellular canaliculus is very short, and it has hardly any microvilli within it. Instead of the microvilli within the cytoplasm, we can see these tubular vesicles. So what happens is that when a parietal cell is stimulated to produce hydrochloric acid, The tubular vesicles that we saw in the resting state, they fuse with the cell membrane, and this is how they form the large intracellular canaliculi and microvilli. And this eventually increases the surface area of the cell membrane for diffusion and ion pumps. So let's talk about the production of HCl, a little bit of biochemistry. Let's look at how this substance is produced by the parietal cell within the mucosa of the stomach. So over here, we can see that within the parietal cell, the very first step which you have to keep in mind in HCl production is that water within the parietal cell is broken into the hydrogen ion and the hydroxide ion. And then you can see that this hydrogen ion is then pumped into the lumen of the gastric gland by a proton potassium pump. So this hydrogen ion has entered the lumen of the stomach. Now, step 3, what happens to the hydroxide ion is that it unites with the carbon dioxide and forms bicarbonate ion. This bicarbonate ion is going to be moved or shifted into the blood vessels, that is the capillaries, right? And over here, you can see in return, chloride will be transported from the blood capillaries into the parietal cell. And as this chloride comes into the parietal cell, it is then going to move from there into the lumen of the stomach. So eventually in the stomach, you've got chloride ions and you've got the hydrogen ions. So they will bond and they will form the hydrochloric acid. So these are the steps involved in the production or secretion of hydrochloric acid by a parietal cell. This is the same thing that I just discussed, but a little more in detail, in which you can see that these here are the intracellular canaliculi of the parietal cell. And these intracellular canaliculi, you can see in the wall of the parietal cell is present the proton potassium ATPase, which is responsible for the secretion or release of the hydrogen ions into the lumen of the stomach. And in exchange for that, potassium is moved from the lumen into the parietal cell and from there eventually into the blood vessels. Also from the blood capillaries, you can see this here antiporter channel, which transmits chloride ions into the parietal cell in exchange for the bicarbonate ions going into the blood capillaries. These chloride ions will then be transported into the lumen of the stomach by a chloride uniporter channel. And here eventually the chloride and the hydrogen ions will unite and form the HCl or hydrochloric acid. Also, you can see in this diagram is the very important sodium-potassium pump or sodium-potassium ATPase. This is responsible, as if you recall from your previous physiology lectures, that it is responsible for moving the sodium out of the cells, whereas the potassium is moved into the cells because of this sodium-potassium ATPase. So for these pumps, energy is required, and that is provided by mitochondria. And this is why when the parietal cell is in the active state, it has a large number of mitochondria. Okay, now we're going to talk about the other cell type which is present within the gastric glands, especially in the fundus of the gastric glands, and they are the enteroendocrine cells. Remember, the enteroendocrine cells are specialized cells which are present throughout the mucosa of the digestive tract, and they are collectively known as the diffuse neuroendocrine system, which has been abbreviated as DNES. That is the diffuse neuroendocrine system. Okay, so the DNES or the diffuse neuroendocrine system. So this is consisting of the cells which are scattered throughout the digestive tract, and collectively they are known as the diffuse neuroendocrine system. The cells of the diffuse neuroendocrine system constitute less than 1% of the epithelial cells of the gastrointestinal tract. And their secretory products have both paracrine and endocrine actions. Now, what do we mean by paracrine actions? This means when the secretory products of these cells have an effect on the cells which are lying close to them. And in case of endocrine action, the secretory products are released into blood and carried away to a distant site of action where the target site is present. So there are many enteroendocrine cells which are present in our human body in locations other than the gastrointestinal tract as well. In the gastrointestinal tract itself, there are many different examples of cells which are producing different hormones. But a few examples that I'm showing over here with you are, one is of serotonin, the substance which is produced by the cells, by the enteroendocrine cells in the fundus of the stomach. This is a substance which increases the gut motility. Then another example of a substance produced by enteroendocrine cells in the pyloric region of the stomach is the gastrin. Gastrin is a substance which increases the secretion of the gastric acid. Now, if we want to classify enteroendocrine cells, we can do this by simply understanding that they can be of the closed or open variety. Now, what do we mean by the closed cell? In this case, the cell is not in communication with the lumen of the stomach, and it releases its secretory product into the blood capillaries, which are present adjacent to its basal region. And from here, the secretory product is then, as I told you, carried away to a distant target site. In case of the open type of enteroendocrine cells, the cell is in communication with the lumen. So the substances which are present within the lumen of the stomach, they are analyzed or sorted out by these enteroendocrine cells, and in response to those substances, in response to the trigger, they then produce and release their secretory products, which are then carried away by the blood capillaries. Now let's discuss the regional differences in the stomach histology. The stomach has different parts. As I showed you before in a simple diagram, that the part which is present adjacent to the esophagus, that is known as the cardia. And then the remainder of the stomach, we can think of it collectively as body and fundus. And then the part which is the lower narrow portion, which is the pylorus. Well, these three different regions of the stomach have a different histology of the mucosa. So how is the histology different? Well, this is a simple table which is showing the comparison between these regions. If we look at the fundus and body, in this part, the depth of the gastric pit is less, and the coiling of the basal region of the gland is also less when we talk about its comparison with the pyloric region. In the pyloric region, the gastric pits are deeper and there is more coiling in the basal region of the gastric glands. And if we look at the cells, then the parietal cells and zymogen cells, they are numerous in case of the fundus and body, but they are very few to absent in case of the pyloric region of the stomach. So these are some essential differences in the histology of the different regions of the stomach. So if you are looking at, suppose, the different micrographs, you can recognize the region from where this micrograph or this section, this tissue section was taken on the basis of these general differences. So you can see over here where my laser pointer is, this is the pyloric region. Here, the depth of the gastric pit is more. And in the basal region of the gastric glands, you can see the increased quantity of coiling. And this is in comparison to the less or shallow gastric pits in the region of the fundus. So remember, in case of pyloric region of the stomach, the pyloric glands, they are deeper, with the deeper gastric pits, so deep that they are extending about half the total length of the mucosa in the pyloric region. Also, there's a special feature in the pyloric region. The muscularis externa is going to be thickened to form the pyloric sphincter in the circular layer. Now we come to the clinical relevance of this topic. I told you before we will be discussing ulcers, which are very important clinically, as we see many people suffer from gastric ulcers. And ulcer can, remember, occur anywhere between the lower esophagus and the jejunum. So we can define the ulcer as a painful erosive lesion of the stomach, which may extend into the deeper layers of the stomach. So if we generally look at an ulcer, it is like a crater. So we can see that it has involved these layers of the stomach wall, and it can also extend so deep into the layers that it can cause a perforation of the stomach wall. Now, the ulcer of the stomach, which is also known as peptic ulcer, is multifactorial. There are many different factors which are involved in causing the stomach ulcer. Let's look at these one by one. I'm going to discuss H. pylori in the last. First, we'll look at the NSAIDs, that is the non-steroidal anti-inflammatory drugs. What are the NSAIDs and what is their role in causing the gastric ulcer? Well, NSAIDs, remember, the examples are ibuprofen, naproxen, aspirin, diclofenac. These are the drugs which can cause gastric ulcers. Why? Because their mechanism of action is that they inhibit cyclooxygenases, the enzymes which are involved in the synthesis of prostaglandins and thromboxanes. So although this is their primary action, that they'll be anti-inflammatory, and they are also going to inhibit the clotting by inhibiting these thromboxanes. So what is going to happen is that the side effect of these NSAIDs could be that they can cause bleeding in the gastric mucosa and also ulcer formation in the gastric mucosa. So other than these NSAIDs, which we should avoid in excessive quantities, and also a chronic overuse, as that can cause the peptic ulcer, there are some factors like stress, smoking, alcohol consumption, and also eating spicy foods. They can all contribute to the formation of a gastric ulcer. Genetics can also be involved. For example, the Zollinger-Ellison syndrome, also known as the gastrinoma. This is a tumor of the gastrin-producing cells, which are present in the region of the duodenum or could be in the pancreatic islets. In this case, there is an excessive amount of gastrin secretion, and gastrin is the hormone which will then stimulate the secretion of the hydrochloric acid, and that in turn can cause the formation of the gastric ulcer. So these are the different factors involved in the peptic ulcer. Now coming to the H. pylori, this was an interesting discovery which was made quite many years ago, that the involvement of H. pylori, it was discovered by these scientists, Warren and Barry Marshall, who in 1982 discovered the association or the role of H. pylori, which is a bacterium, in gastritis and peptic ulcers. So previously it was thought, before the discovery of the role of H. pylori, that there are multiple factors which are involved in the formation of peptic ulcers, but the role of bacteria was not considered to be a causative factor, because it was generally supposed by the scientist community that because of the acidic environment of the stomach, which is produced by the hydrochloric acid, it is not possible for bacterium to survive within this harsh environment. But this was challenged by the scientists Warren and Barry Marshall, and these scientists, they played an interesting role in the discovery of this H. pylori, in that Barry Marshall self-inoculated himself with the strain of the H. pylori, which then, by ingesting the H. pylori, and that actually produced gastritis and gastric ulcer within the scientist himself, Barry Marshall, and then he treated himself with the antibiotics, and the ulcer was then treated. And this proved as an essential contributory factor for determining the role of H. pylori in the production of the gastric ulcers. And it also showed us how the use of antibiotics can then help in the treatment of the gastric ulcers. So if you're interested in about reading up on that history, I've shared this link for you, which is an article on PubMed. If you're interested in the discovery of H. pylori and its relevance in gastric ulcers. Anyhow, coming back to our topic, when we are talking about H. pylori, we are going to discuss why is it so resilient? How does it resist the bacteriostatic effects of hydrochloric acid? Well, the H. pylori has an enzyme which is known as urease, and this is the enzyme that hydrolyzes urea to form a protective ammonia cloud, and that makes this bacterium resistant to the acidic environment of the stomach. So the ammonia cloud acts as the shield for the bacterium which has this enzyme urease. The lipopolysaccharide antigens expressed on the surface of this bacterium, H. pylori, also remember they mimic, or you could say they resemble those of the gastric epithelial cells. So the antibodies which are produced against H. pylori, they also damage the gastric mucosa. So this is an additional effect. This is an additional factor which causes the gastritis and eventually leads to the formation of the gastric ulcer. Nowadays, it is quite easy to diagnose the H. pylori infection. There are different methods, there are different tests that can be used. The blood test can be used, the stool test can also be done, but here is the very simple, painless, non-invasive procedure, which I've shown over here is the urea breath test. This is a rapid and accurate method which takes just about 20 minutes, in which the patient is required to swallow a capsule which is containing urea with the radioactively labeled carbon. And if, let's suppose, the bacterium is present, then it would have within the stomach, H. pylori, as I told you, has urease enzyme. It would then dissolve that urea into ammonia and carbon dioxide. So after a short while, the patient is asked to exhale into an airbag. And if that air contains the carbon dioxide with the radioactively labeled isotope carbon, the one which was present in the urease, which was present in the urea. So that then confirms that urease enzyme was present in the stomach of the patient, and that means the presence of H. pylori, that is Helicobacter pylori, is confirmed, because it would then have broken that urea into ammonia and carbon dioxide with the radioactively labeled carbon. So this is a simple test, can be done. Otherwise, the endoscopy and the stomach tissue biopsy can also be taken for directly assessing the presence of looking for the presence of the H. pylori within the gastric mucosa. Okay, so once we have confirmed the presence of the ulcer, and also we have confirmed the presence of H. pylori in relation to the ulcer, what is the treatment? Well, you know that generally speaking, proton pump inhibitors are advised for gastric ulcer, like omeprazole, esomeprazole. Also, the histamine receptor antagonists, which are ranitidine, cimetidine, they can be prescribed, as this would also inhibit the production of HCl. Antacids also give certain relief. These are the drugs which neutralize the acidic effects of the gastric juices, such as bismuth, magnesium, calcium salts. And the antibacterial drugs like amoxicillin and clarithromycin or metronidazole, they eradicate the H. pylori. And triple therapy, this is a standard regime, which is the triple therapy, in which 2 antibiotics and a proton pump inhibitor are prescribed to the patient. And this is the first line of treatment in peptic ulcer, which is due to the H. pylori infection. But remember, this is, of course, a very resilient organism, and there are many recurrences which take place, and therapy needs to be prolonged over a longer period. Okay, so now that we have discussed the basic histology and also the clinical aspects of the histology of the stomach, now we're going to have just a quick look at this slide, which is going to show you the glands. So I'm sure you can identify these cells over here. These are the parietal cells and the mucous neck cells over here in the gastric glands. And then if we look over here, we can also see these chief cells or the zymogen cells. And between the gastric glands, remember, we can see the lamina propria in the transverse sections in the histomicrographs. So this concludes with today's topic, which was histology of the stomach, in which we talked about the different cell types which are present and within the stomach mucosa and the different parts of the gastric glands. And in relation to that, we also discussed the gastric ulcer and how we can understand the association of Helicobacter bacterium with the gastric ulcer and the different methods of treatment of the gastric ulcer. So I hope this was an interesting topic for you and you got all the essential points of this topic. This is very important when it comes to exams, and it's also important with the clinical relevance. So if you like this lecture, if you found this video presentation interesting, please like and subscribe to my YouTube channel, which is AnatomyMentor. You can simply do that by clicking on the picture that is going to be appearing at the end of this video. And please also just press the bell icon so you would be notified with my video uploads. So thank you for your time and take care and enjoy your studies till we meet again. Goodbye.